സമാധാനത്തിലെടുക്കാം. Hello. Hello. Am I audible? Okay. So, my dear students, again I am here back. Amala Anjos. I am a biology faculty of Goscore Learning. So, welcome to Goscore Learning, GCC's fastest moving edtech. So, my dear students, today I am here with the another chapter of your biology textbook, that is biotechnology, chapter number 9, biotechnology principle and processes. Okay. So, in this chapter, you have to be very much thorough about the previous chapters, that we have already discussed already. That is which chapter, molecular basis of inheritance. In that chapter, you have learned what is DNA, what is the structure of DNA, and how the DNA is formed between the nucleotides, right? So all those concepts are very important to study this biotechnology principles and processes, right? So this is a particular a separate wing of biology, actually. Okay, biotechnology, you can integrate the word biotechnology, right? Bio and technology, when bio meets the technology, it forms biotechnology. So let's see what are the sorry, what are the different contents which is present in this chapter. And this chapter you have to study about what are the different types of biotechnology, where this biotechnology will be dealing upon, and what are the different tools for biotechnology, then what are the different steps in biotechnology. You have to learn about gel electrophoresis, then we have to study about PCR vectors, different things we have to learn about. Okay, so let's start our chapter. So this is, as I have told, that this is chapter number 9. It is very important. Okay, 6 marks will be coming from this chapter. Okay, from biotechnology 1 and 2, it will be having 12 marks. So my dear students, we can predict at least minimum of 6 marks or 9 marks from this chapter. Okay, so this is the chapter, that is biotechnology principles and its processes. So let's learn what is biotechnology. We have to define biotechnology, right? So let's define what is biotechnology. It is the technique of using, okay, it is the technique of using a live organism. It is the technique of using live organism or enzymes from organisms to produce products and processes useful to humans. See, this is the technology where the microorganisms have been used to produce the useful substances. For example, in the previous years, or in previous uh, ages, what we can say, all ages, we not only in all ages, in new era too, we are using the substance called curd, right? Curd. What is, how this curd is formed? See, we can take milk. Okay, we can take milk, and this milk is converted into curd in the presence of an organism. In the presence of which organism? It is lactobacillus. Okay, which organism it is? Lacto. Okay, milk, it is converted into curd in the presence of lactobacillus. So we can say this lactobacillus is a bacteria. What is lactobacillus? It is a bacteria. So we can, once again, we can check the definition. See, it is the technique of using live organism. Here live organism has been used. What is the live organism? Bacillus. Okay, and or enzymes from the organism to produce products and processes. Okay, to produce products and processes, what product is produced? Curd is produced to which is useful to human. We human beings are using curd in our diets. Okay, so we can integrate our definition with this example. Okay, this was a previous technology and it has taken part in these days also. Along with this, a new modern technology has been arised. Okay, in this new modern technology or biotechnology, what we are using same thing, that is the live organism. Okay, sometimes live organism has been used, or the enzyme which is produced by the organism has been taken. Producing enzyme means the gene is taken out and this gene is inserted into the host organism with the help of a vector organisms. Okay, so though by producing such a product, what is the end result? The end result will be very much useful to the human beings. Okay, is it clear for you? So that's all about the biotechnology definition or definition for biotechnology. So next one we have to learn about what is the important union or federation of biotechnology. Okay, who is the important federation? The European Federation of Biotechnology is the important group which gives some definitions for biotechnology. Okay, and also this particular federation will decide the approvals of different biotechnologically processed life organisms or products. Okay, so let's see how they have defined biotechnology. Okay, see, it is the integration of natural science. Okay, it is the integration of natural science and organisms, cells, parts thereof. Here different things have been used. Okay, it is the integration of natural science. Okay, integration of natural science along with how it is integrated. It has been integrated with organisms, different organisms. Conditions have or different steps is there, that is either it will be organisms. Okay, complete organisms will be taken, or cells will be taken, or parts of the organism will be taken. Okay, parts of the organism will be taken, and molecular analogues of products and its services. So by using or integrating everything this with this, what is the result? We will get a result as a product. Okay, and this product has been taken, and this product has been taken for the human beings, for the needs of human beings. Okay, that's all about the in definition for biotechnology. Okay, so let's see what are the different types or modern biotechnologies principles. Two different principles. What are the two different principles? Okay, the first principle is genetic engineering. Okay, the importance of biotechnology lies here, that is genetic engineering. Second one, it is chemical engineering, also we will call it as biochemical engineering. Okay, it is also known as biochemical engineering. Okay, what is the result of these genetic engineering and biochemical engineering? Okay, genetic engineering, you know what is about gene, right? What is gene? What is gene? Gene is a part of DNA. Gene, it is a part of DNA, and also this gene defines the characteristic phenotype of a person. Okay, so we can say this genetic engineering is the engineering of genes. Okay, and also you might have studied that genes have been inherited from one generation to next generation. So the engineering of genes which is present inside the human beings for getting a new enzyme or to produce a new enzyme or to modify the structure of enzyme is known as genetic engineering. So let's see, in genetic engineering, it is defined as the direct manipulation of genome. Okay, what are the different genomes you have learned in the previous classes? That is DNA and RNA. What is DNA? Deoxyribonucleic acid. Okay, what is RNA? RNA is ribonucleic acid. So, if there is any kind of manipulation in this DNA and RNA, the result of mRNA will be changed. Okay, if the mRNA is changed, what happens? The enzyme produced will be also changed, or the polypeptide produced after the translation will be also changed. So we can say the engineering, it is the direct manipulation of DNA or RNA of an organism. It involves the transfer of new genes to improve the functions or trait into the host organism. So why we are taking this or manipulating the genes of an organism? It is manipulating in order to get a better result. Okay, in order to get an improved or to improve the function of that particular trait. For example, we can say we have black eyes, right? We have black eyes, and this black eyes is determined by the genes present in our body. So if we are taking this genome, and if you are manipulating this with the other colors, okay, then the color will be changed into brown as per our desire. Okay, and also next example, okay, we can look into the plants. Okay, so plants produce in a minimal quality, the products is taken, and if you want to manipulate, if you want to get a more product from this plants, what we are doing, we can take the better genes from different plants, and we can integrate the same genes into the host organism, so that a better result or better yield can be taken. Okay, that kind of process is known as genetic engineering. So we can say the wing of biotechnology comes under this genetic engineering. Okay, next one, it is chemical chemical engineering. What is chemical engineering? It is the maintenance of sterile condition in chemical engineering process to enable growth of only desired microbes. Only the desired microbes will be manufactured. Okay, and also in this biochemical engineering, what are the products in biotechnology after this biochemical processing? The products are very important. Underline this and study. Okay, what are the different products? It is antibiotics, vaccine, and enzymes. Okay, once again, what are the products after biochemical processes? It is the products which enable the growth of organisms, that is, organism or microbes, desired microbes, for the manufacture of different chemical substances like antibodies, then vaccine, enzymes, etc. Okay, clear? Fine. Next, we can say what are the, now we are entering into the core of the chapter, okay? Core part of the chapter. That is, first one, what we have to learn? We have to learn about the steps of recombinant DNA technology. My dear students, before entering deep into this chapter, we have to know about what are the different steps that is taking part in biotechnology. What are the different steps? First, we can say if there is a desired gene, that gene can be taken out. Okay, cutting. Okay, that is isolating the genes. Then if you are looking into this isolated gene, see a small isolated genes have been formed. This isolated gene can be integrated into a vector organism. Into a vector organism. That is simply we can say cutting and pasting. Okay, cutting of gene and it is integrated into the genome of other organism. That is the basic principle behind the very basic principle behind the biotechnology. Okay, so that concept should be there in your mind. Then let's see what are the different steps involved in biotechnological processes. Okay, first one, identification of gene or identification of DNA with desirable genes. For example, if there is a plant, okay, this plant is very short, okay, but there is another plant of same type, of same species, which is very tall and it produce more yield than this short plant. Okay, so what is the condition here? We have a long, tall, and the genes present inside this plant will is decide its height and its yield. Okay, the gene is taken for height and yield. So our desired trait for the gene is height and also the yield. So this genes from this plant will be taken and it is given to another organism for its multiplication, that is vector organism, and later on it is given to the this plant, and finally we will get a result from that plant. So it is important to understand what kind of gene have to be taken out from the plant. Okay, that important plant we can say, or that important character we can say, that is our desired gene. If desired gene is chosen, this desired gene can be integrated with the other host genomes. Okay, so that is the first step. Once again, what is the first step? Identification of DNA with desirable genes. What is the second step? It is the introduction of identified DNA into host cell. As I have mentioned, if DNA from this part is taken, then it can be given to the host organism. In between, there are different steps. Okay, you have to study about that too. Okay, so third one, it is maintenance of introduced DNA in the host and transfer of DNA to its progeny. Okay, if you understand this concept, see the desired gene height yield, it is that is the desired gene of this plant, from this plant. It is then given to this is the host organism, that is the host plant. Okay, so if it is taken and if it is given to the plant, it has to be maintained inside this plant. Okay, the short DNA has to be maintained in this plant. And also, my dear students, this in this plant we are integrating this gene, right? So from here it has to it should be able to transfer the gene to. Okay, that is the second point. The third point, that is transfer of DNA into its progeny. Okay, so this is the basic first step. Questions will be asked. What are the different steps involved in recombinant DNA technology? Tongue slip. Okay, don't mind. Okay, so fine. So let's see about this in detail. The same thing that we have mentioned. First one, identification of DNA with desirable genes. Okay, what is given here? Traditional hybridization techniques leads to inclusion and multiplication of undesirable genes and desirable genes. So that means normally we have taken the hybridization techniques, right? So during hybridization we know that cross pollination will be taking place in the case of plants. Okay, so during cross pollination all the characters will be mixed. All the characters, undesired characters also will be taken, and it will be also passed to the next generation. But if you are considering into this particular biotechnology, we can say only the desirable genes can be transferred from one generation to the next generation, and also from the trait taken from the plant and to the next host organism. Okay, so in genetic engineering, only desirable genes. See, it is very important. That term is very important, dear students. It should be there in your answer sheet. Okay, what is that? In genetic engineering, desirable genes are introduced. What are introduced? Desirable, the needed gene. Only the wanted genes are taken and it is transferred from one plant to the next plant. Okay, clear? Fine. Okay, that's all about the identification of DNA with desirable genes. The second important point or second important step involved in recombinant DNA technology is introduction of identified DNA into the host. Okay, what is introduction of identified DNA into the host? See, we have identified a particular DNA. Okay, that DNA has to be transferred into the host organism. So how it can be transferred? Here another important term is present, that is vector. Okay, a vector DNA such as plasmid is used to an alien piece of DNA into the host organism. So first we can say the desired gene is taken and it is then given or integrated to the vector or plasmid. Okay, usually vectors are plasmids of bacteriophages. So we can say if this is the desired gene, it is then integrated to a plasmids. Let's say it is integrated to a plasmid or vector genes. Okay, vector. Then later on, this vector genes or plasmid genes is given to the host organism. It is given to the host organism. Let's say any plants. Okay, or any animals. What is the host organism there? It is given to the host organism. Is it clear? These are the three different steps involved in recombinant DNA technology. Okay, fine. If it is fine, you can give me thumbs. No thumbs are getting these days. Okay, so fine. Let's go to the next topic. That is next step, maintenance of introduced DNA in the host and transfer of DNA into its progeny. So once it is entered into the host cell, as I have mentioned, it should be retained. Okay, it should be maintained, and it should be able to replicate in its next coming generation. It is given here. Let's see. A piece of DNA has no sequence called origin of replication needed for the starting of replication. So it cannot multiply itself in the progeny cells of the organism. Hence, alien DNA is integrated into the recipient genome. It multiplies and inherited with the host. Okay, as I have mentioned earlier, first it has to be taken out. It has to be given to the vector DNA. Okay, then this vector DNA is given to this host organism DNA. Later on, the host DNA and the alien DNA will be integrated together to function in a proper manner, and it will give a it will give a result, or it will give an end product which is very much beneficial for the humankind. Okay, clear? Fine. Bye. Okay, so that's all about this particular three different steps. Next, we have to study about what are the different tools of recombinant DNA technology. My dear students, these are very important. Okay, these are very much important. Among this, very much important is restriction enzyme. Okay, it is the restriction enzyme. Next, we have to study about polymerase enzymes, ligases, vectors, and host organism. We have already known, or we know about polymerase enzyme, right? Polymerase enzymes are the enzymes which is used in both replication and transcription. In replication, it is DNA polymerase, whereas in the transcription, it will be RNA polymerase. Different RNA polymerase is there. RNA polymerase 1, 2, 3. Okay, so we can say in this case, it is regarded with tag polymerase. Okay, tag polymerase is being used in PCR, polymerase chain reaction. We'll learn in later in coming session. Okay, so fine. Next one, ligases. Ligases are also the enzymes which is used to combine two different DNA. Okay, see, you know about the Okazaki fragments which is produced after DNA replication, right? So there, what is coming and joining the DNA? We have ligases to join the DNA in between the Okazaki fragments. So same ligases have also been used here for the process of biotechnology in different stages. Let's see in which stages that is being used. Okay, next, vectors. Vectors, we know what are vectors. See, if you are considering the case of malaria, this that is a disease which is caused by a causative organism, and it is carried by, actually it is carried by a it is carried by the mosquito. Okay, so there the mosquito is acting as the vector, and it is spreading to all of the humankind. Okay, so there we can say the vector is acting as, or the mosquito is acting as the vector, and we can say the parasites are entered into the organism, host organism, with the help of a vector organism. The same concept is also being placed here about vectors, that the gene present inside, the desired gene is taken out of the organism, and that desired gene is given to the vector organism. Later, this vector organism is its DNA or the plasmid is given to the next set of that host organism. That concept is being done here exactly with that of the example for malaria. Okay, so we can coordinate and study. Okay, next, last one, it is host organism. Host organisms are the organism where the trait has to be expressed. Okay, when the trait is expressed, we will get a proper result from that particular host organism. Okay, so these are the different tools for recombinant DNA technology. Okay, so next we have to study about the restriction enzyme. Very important in this chapter. Okay, this is very important, means very important. Why? Because every year from this part there will be for sure a question will be from this side. Okay, so it is very important to study this particular point. Dear students, when you're going for examination, you have to be thorough with restriction enzymes. Okay, so see, my dear students, what is restriction enzymes? That is a question, right? So this restriction okay, restriction enzymes They are also known as molecular scissors. What they are known as? They are also known as molecular scissors. What is molecular scissors, or what is the function of scissors? Dear students, we know the function of scissors, right? If there is a piece of cloth, the function of scissors is to cut. Okay, the function of scissors is to cut. Then what is the function of molecular scissors or restriction enzyme? Restriction enzymes are the enzymes which are used to cleave the DNA. Okay, so we can say these restriction enzyme belongs to a larger class of enzymes, that is nucleases. Okay, they belongs to a larger enzymes. You know what is the function of enzyme, right? Enzyme is the molecule which increase the rate of reaction in biological reactions. Okay, so this enzyme, restriction enzymes, are the enzymes which is belonging to which class? They are belonging to nucleases. You know different classes, we have already studied in grade 11 about different kinds of enzymes, right? Oxidoreductase enzyme is there, isomerase is there, ligase is there. Different kind of enzymes and enzymatic reactions we have already done in grade 11. So here what we have to learn, in which class this restriction enzyme belongs to? Restriction enzyme clearly belongs to a large class, that is nucleases. So my dear students, we can say the nucleases or the restriction enzymes can be restriction enzymes can be of two different types. Okay, restriction enzymes can be of two different types, that is exonuclease. Okay, exonuclease and endonuclease. Exonuclease and endonucleases. So from the term itself, you have to understand what is exonuclease and what is endonucleases. Dear students, exo means it is the part or it is the type of restriction enzymes where the DNA is cut at the ends. Okay, if the DNA is cut at the ends, we can call that kind of restriction enzymes as exonucleases. Next one, endonucleases. Endonucleases are the nucleases that are being cleaved at the inner sides of the DNA. Okay, that is the meaning of endonucleases. Let's see the definition for both exonuclease and endonucleases. Okay, so in the case of exonucleases, see exonucleases, it removes the nucleotides, not sides, okay, it removes the nucleotides. We have learned what is nucleotide, right? So if you are considering a sugar, a DNA, there is a pentose sugar. If there is nitrogen, there is a nitrogen base, and this is a phosphate group. In this position, there is phosphate group. Okay, so if it is present like this, we can say this is a nucleotide. Okay, this is a nucleotide. So in the case of DNA, it is double stranded. In the case of DNA, it is double stranded, that is adenine pairs with thymine, guanine pairs with cytosine, right? So we can say this is a kind of DNA where nucleotides are present. Okay, where nucleotides are present. Here we can draw 3 nucleotides or 4 nucleotides. Okay, so see, nucleotides will be present inside. So what is the function of exonuclease? Exonuclease removes nucleotide from the ends of DNA. That means you can find the ends here. This is one end and this is the another end. So this exonuclease will be acting upon this end of DNA and it will cleave the DNA. Okay, it will cleave the ends of DNA and then we will get a result. We will get a whole result. Okay, so what is next one? It is endonucleases. What is endonucleases? If you are looking into endonucleases, see, let's draw an DNA. Okay, this is another DNA. Okay, so what is, if endonucleases are present, endonucleases can cleave in the inner sides. Okay, it can be cleaved in the inner side so that a product will be given. So two types of ends will be produced, sticky end and blunt end is formed. Blunt end is formed in the case of exonuclease, whereas sticky ends are produced in the case of endonucleases. Okay, that is the difference, major difference between endonuclease and exonuclease. Dear students, once again I'll repeat. In the case of exonuclease, the resultant DNA will be having blunt end. Okay, it does not have a sticky end. Whereas in the case of restriction endonucleases, it will produce the sticky ends. Okay, let's see what is sticky ends. Okay, and also see, endonuclease makes cuts at specific position within the DNA. For example, the first restriction endonuclease. What is the first restriction endonuclease known as? The first restriction endonuclease is also known as HIND2. Okay, the first isolated endonuclease, sorry, endonuclease is HIND2. Okay, and what is the function of this HIND2? It always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. If a sequence of 6 base pairs are present in a particular area of DNA, at that particular site, what happens? This particular endonuclease will go and make a cut, and it forms a sticky end. Okay, the specific base sequence is known as recognition sequence. It is important. What is recognition sequence? Okay, the sequences which is recognized for the cleavage of DNA. Okay, usually in the case of exonuclease, it will be present at the ends, but in the case of endonuclease, it will be present at the inside the DNA. So we can say, what we can say, the endonucleases or exonucleases have a specific number of sequences to identify the cleavage site, and that particular point is known as recognition sequence. And this sequence, 6 base pair sequence, okay, that 6 base pair sequence present, 6 base pair sequence in the DNA. Okay, if 6 base pair sequence is present in DNA, what happens? That will be acting as the recognition sequence, and if recognition sequence are identified, then the restriction endonuclease will act upon that particular 6 genes, and it is cleaved. Okay, as a result of endonuclease action, what is produced? Sticky ends are produced. Please note it down. Okay, it is very important. What ends are produced? If endonuclease is acting, okay, if endonuclease is acting, what is the result? Blunt end will be the result. Okay, sorry, not blunt, sticky ends will be produced. Okay, what ends will be produced? Sticky ends will be produced. If once again, I will write it in another sheet so it is easy for you. Okay, so let's say what are the two different types? It is endo and exo, right? Exonuclease is present and endonuclease is present. Okay, what is exonuclease? Exonuclease remove the base pairs at the ends. Okay, remove base pairs at the end. Remove base pairs at the ends of DNA. Okay, whereas endonuclease, what is the function of endonuclease? It removes the base in within the DNA. Okay, it makes a cleave in within the DNA. Okay, so there will be two kinds of end. What are the two different types or kinds of end? This will be produce a blunt end DNA. Blunt end DNA. Okay, whereas in the case of endonucleases, it will be producing what it has produced? Sticky end DNA. Sticky end DNA. Okay, so is it clear? So we have to study what is sticky end. Okay, what or how sticky ends are produced. See, this is addition of an restriction endonucleases. So see, the enzymes cuts both the DNA strands on the same. This is a vector DNA. Okay, this is a vector DNA and this is a foreign DNA. So endonuclease have to be act upon this. Endonuclease have to be act upon this particular gene. Okay, this particular gene where vector is also present and foreign DNA is also present. Okay, here which enzyme, which restriction enzyme is acting upon? The restriction enzyme acting upon is ECO R1. ECO R1. Okay, eco R1. So when the restriction enzyme eco R1 is acting upon, it cleaves in a particular manner, so that what is produced, a sticky end, both the sticky ends are produced. See, AATTC is removed from here, and from here CTTA is removed. Okay, this is the way how endonuclease will be acting upon when this endonucleases are produced. Okay, not endonuclease, when this DNA is produced after the action of particular restriction enzyme, sticky ends are produced. These sticky ends can be then joined together. See, DNA fragmentation, join at sticky ends. Okay, these are the DNA. This is a vector DNA and this is a foreign DNA. So vector DNA and foreign DNA can be combined together. So a recombinant DNA is produced. What is produced? Here the vector DNA is also there and also foreign DNA is also there. Okay, vector is present and also the foreign DNA is present. Okay, so vector DNA and the foreign DNA combines together to form what DNA? Recombinant DNA. Okay, recombinant DNA. Vector DNA and foreign DNA combines together to form a recombinant DNA. Is it clear? This is about the restriction enzyme. Let's move back how the restriction enzyme work more efficiently. That is about the palindrome sequences. Have you heard about palindrome sequence? Okay, each restriction endonuclease recognizes a specific palindrome nucleotide sequence in the DNA. Have you heard about Malayalam? Right? Malayalam. We will write Malayalam here, okay? All Malayalis come together. Okay, Malayalam. Okay, this is the sequence or this is the letters. Okay, so see Malayam. Okay, so this is one way how we can read in the forward direction. If it is read in the backward direction also, how to read this? How to read this? Malayalam itself. Okay, Malayalam itself. It is that sequences are known as, that kind of sequences are known as palindrome sequences. Okay, so in the case of one palindrome sequence is given here as GATCC. Okay, so let's see. G A T T C. Okay, it is a palindrome sequence where this is 5' end and this is 3' end. Let's read it backwards. Cytosine, it can pairs with guanine. Okay, thymine, it can pairs with adenine. Okay, here one more A is there. Okay, GAATTC. So thymine, it can pair with adenine. Thymine, it can pair with adenine. Adenine pairs with thymine. Adenine pairs with thymine, and guanine pairs with cytosine. See, cytosine, thymine, adenine, guanine. Okay, so we can say palindrome sequences are the group of letters that form the same words when read both forward and backward direction. Example is given as Malayalam, as I have told before. Here, in the case of Malayalam, it is Malayalam, it is given. So, my dear students, it is actually Malayalam, okay? Malayalam. Okay, and also what is given here? Given here is GAATTC. What is GAATTC? GAATTC codes for C. Pairs with guanine, thymine pairs with adenine, thymine pairs with adenine, adenine pairs with thymine, adenine pairs with thymine, and guanine pairs with cytosine, which we can read it back as palindrome sequence. Okay, in the case of biotechnology, what is a palindrome sequence? What is a palindrome sequence? Palindrome in DNA is a sequence. You have to study the definition, okay? Palindrome in a DNA is a sequence of base pairs that reads same on two strands. Okay, that reads same on two strands when orientation of reading is kept the same. The orientation, when the orientation is kept same, how can we read? We can read in the same way for the two strands. Okay, that is palindrome sequence. When palindrome sequences are present, what acts endonucleases can act on. Okay, clear? I have shown you, right? See in the previous picture that we have learned. Okay, see here it is given GAATTC. So here also CTTAAG, that is GAATTC. Okay, this is the sequence in vector DNA, whereas in the case of foreign DNA, what is the sequence? There also the sequence is GAATTC. Here GAA DTC. So the endonuclease, restriction endonuclease, both in both the case of vector DNA and in the foreign DNA, what can act? The endonuclease can act. Example, EcoR1. Okay, EcoR1 can act upon both this vector DNA and also foreign DNA. When it is acting upon the vector DNA and foreign DNA, what happens? The sticky end is produced. Sticky end means the end which can be sticked together. Okay, that is a simple meaning. Okay, sticky ends are produced. When sticky ends are produced, what happens? Recombinant DNA is formed. Okay, clear. Next, my dears, we have to study about the nomenclature of restriction enzymes. Okay, so restriction endonucleases, particularly we can say restriction endonucleases. For example, we can say EcoR1. Okay, EcoR. Okay, it is EcoR1. Okay, so this is the name of one particular restriction endonucleases. Here, what does this E represent? What does this represent? CoR1 represent? This we have to learn. So, this particular restriction enzymes is actually taken from bacteria Escherichia coli. Okay, Escherichia coli. So, E represent, what does the E represent? E represents the genus name. Sorry, genus name. E represents the genus name. CO represents the species name. Okay, co represent the species name. And next one, what is the next letter? See, the next letter is R, right? R represent the strain. Okay, R represent the strain of bacteria from where this is isolated. Then last one, it is first one, it is given. Here it is one given, right? So this one represents the order of isolation. Okay, the order of isolation. Order of isolation. Okay, so it is also very important. You have to study the example with eco R1 hind 2. Okay, so it does not mean how much it is isolated or first one is isolated. It just represents the order of isolation. That means the Roman number. Okay, this is very important. E represents the genus name of the particular organism from where this restriction endonucleases have been isolated. Okay, co the species name. Okay, then R strain. Okay, I order of isolation. Okay, that's about the name nomenclature of restriction enzyme. Clear, right? Is it clear? So one more image we have to study in that particular point, that is about this one. Okay, the same, the functioning of restriction endonuclease is clearly given here, and also how the genes have been uh, integrated. Okay, so see, this is a foreign DNA. This is a foreign DNA. Okay, and this foreign DNA and this foreign DNA. Okay, this is a foreign DNA, and here you can find the vector DNA. Vector DNA, we can call it as plasmid also. So what is plasmid, my dear students? Plasmid, which is present, an extra chromosomal DNA which is present in bacteria, is known as plasmid. Okay, so we can say from both this DNA, what happens? Restriction enzymes will be cutting the DNA and vector DNA at specific point. If the sequence palindrome, see, if endonuclease is acting, the palindrome, if the palindrome sequences are present, then at that particular site the DNA will be cut. Okay, we can say DNA will be cut. Okay, and after that what happens? See, a cut ends are formed. The small portion of the cut ends, here you can find the small portion of the cut end, right? The small portion, it can be at the same time here also the palindrome sequence will be present, and it will be also cut down. Okay, and this, once again. Mm. Okay, so this one, in this case, what happens? The small part of the DNA, small part of the gene, it is taken and it is given to the, which part? It is given to the that particular foreign DNA. Okay, that integration will be taking place. The small part of DNA, it is taken and it is then integrated to the this particular vector DNA. Okay, this we can predict a question from this particular figure. Okay, in this figure also, and from the previous figure, we have to understand what kind of enzymes are used. See, in the first case, this is foreign DNA and this is vector DNA. Study that it is foreign DNA and this is vector DNA. Okay. Later on, what happens? We want to cut this both the DNA, right? If you want to cut the DNA, we have to use the restriction enzyme. Here, what enzyme is used? We can say restriction endonucleases have been used. Okay. If restriction endonuclease have to be acting upon, what is the condition? The condition is, what is the condition? The condition is it should possess a palindrome sequence. Okay. So we can say these possess a palindrome sequence or restriction enzyme cut. Okay, restriction enzyme cut. So after this, what happens? The small part of the DNA is given or ligated. Okay, the small part of the DNA is ligated. See this blue color, small color DNA, it is ligated to this region. Sorry. Okay, it is ligated. Where after ligation, see, ligases join foreign DNA to plasmid. Okay. Okay. So then what happens? Transformation. It is integrated to a again it is integrated to a DNA E. coli, from where the E. coli cells or the vector DNA will be multiplicated. Later on, this DNA can be taken into and given to the host organism. Okay, so this is an important question. Restriction enzyme is an important question. In restriction enzyme, palindrome sequence is an important question, and you can find what are the different types of restriction enzymes. Okay, endonuclease, the types of restriction enzymes, right? So endonuclease and exonuclease, every year they have a tendency to ask the question from this particular portion. Okay, so it is important. You have to study that. Okay, about clear about this? Okay, fine. So next one we have to go with cloning vector. My dear students, what is cloning vector? I have mentioned, okay, right? I have already mentioned. See, we are taking some DNA from the organism. The desired trait is taken from a particular organism. Okay, so that desired DNA have to get integrated with a particular DNA so that it can be given to the host DNA. Okay, that is the condition. So what happens in the cloning vector? In cloning vector, we are taking the desired DNA, cutting with a restriction enzymes, and the DNA is given to the foreign DNA or the vector DNA. Okay, so let's see what is a vector, a cloning vector. Okay, or what acts as the cloning vector. Here, plasmid, my dears, what acts as the important. Okay, plasmids. Okay, this is plasmid and bacteriophages. Okay, plasmid and bacteriophages, both of them is commonly used vector for cloning. Okay, what is cloning? I told you, the cloning is giving a desired DNA to this plasmid DNA is known as cloning. So we can say for cloning, what are the DNA used? The DNA used is plasmid and bacteriophages. Again, I am saying, what is a plasmid? What is a plasmid? See, you have studied the structure of bacteria, right? Inside bacteria, bacteria are normally, they are prokaryotes. Okay, they are normally prokaryotes. So chromatid will be present. Okay, what is present? Chromatid will be present. At the same time, there will be a circular extra chromosomal DNA. Okay, there will be a circular shape, a circle in shape. Okay, that circular extra chromosomal DNA is known as plasmid. We are taking out that plasmid and integrating the desired DNA into the plasmid. Okay, so bacteriophages. What are bacteriophages? Again, the question comes, right? So those viruses, okay, those viruses that can be able to integrate into a bacterial body is known as, into a bacteria. Okay, into a bacteria is known as, is known as the bacteriophage. Okay, in the case of bacteriophage, only the RNA, which is only the genetic material present inside, it will be moving into the bacterium. Okay, so we have to understand what are acting as vectors. Okay, clearly study that the vectors are plasmids and bacteriophages. Okay, they have ability to replicate within bacterial cells independent of the control of chromosomal DNA. What does it means? As I have told my dear students, in the case of bacteria, see, in the case of bacteria, let's do it in another. Okay, see, this, if this is a bacteria, if this is a bacteria, inside the bacteria, what is present? Chromatin is present, right? It is prokaryote. Since it is prokaryote, we can say chromatin is present. And this, there is a plasmid. Extra chromosomal DNA is present. Okay, so if you are integrating the particular gene, okay, if you are integrating the particular gene in a small portion here, okay, in, we can say, in, see, in plasmid, the desired gene is integrated into the plasmid. What happens? It is replicating independently. The plasmid is replicating. Okay, it gets replicated independently. Okay, independently. That means here the replication, here the chromatin is present, right? Here the replication will be independent. Okay, it will also get replicated, but it is not affecting anything regarded this plasmid. Okay, clear? That is given in that particular sentence. They are independently replicating. Okay, then bacteriophages, because of their high number per cell, have very high copy numbers of the genome within the bacterial cells. Since bacteriophages can be multiplied very soon, bacteriophages can also be considered to be a vector. Okay, clear? I think it is clear to you. Okay, so next one, what are the requirements? Okay, what are the characteristic features which is facilitating the cloning vector? Okay, what should be there inside the cloning vector? Okay, that is also another concern. What is there? What should be there? It should be origin of replication. Dear students, we have learned what is origin of replication in our chapter. Which chapter? Replication, molecular basis of inheritance, right? So, if there is a DNA, particularly in the case of a bacterial DNA and plasmid, see, when does when does when does the replication starts? Okay, the replication have to be started only with a particular region known as ori region, that is origin of replication, ori region. Okay, ori region means origin of replication, where replication starts. Okay, then only, see, I have told you we are integrating the genes in plasmid, right? So the plasmid has a ori region. That ori region will be helping the plasmid to replicate again and again. So the vector should have which side? Origin of replication. That means the sequence from where replication starts, and away piece of DNA when linked to the sequence can be made to replicate within the host cell. Okay, when it is given to the host cell, it also should be replicated. So we have to understand that the vectors, okay, vectors should have the ability to replicate. If it is replicated, then it can move to the next generation. Simple, that's it. Okay, fine. Understood. The sequence is responsible for controlling the copy number of linked DNA. Okay, so if ori region is present, as I have told, that what happens? Replication takes place. Okay, if replication takes place means replication is the process which is taking just before the what we can say, cell division. Okay, it is the process. See, we have learned in cell cycle, right? In the last chapter, we have learned different phases. S phase, okay, G1 phase, S phase, G2 phase, and M phase. These are the different phases which is involved in cell cycle. Okay, so in the case of S phase, what happens there? The replication takes place. Okay, replication takes place, and in M phase, the cell division takes place. So it is important to produce the DNA before the cell division. So we can have to understand before the cell division, what if the cell division have to be taken, what should be there? The DNA content should be also divided. Okay, so that in S phase, what happens? Replication should be taken place, and for the purpose of this replication or the content to be increased, what should be there? Ori region should be there in the plasmid. Okay, Clear? That is the first point. Then next one, it should have selectable markers. Okay, selectable markers. What is the function of selectable markers? Selectable markers helps in the identifying and eliminating non-transformants and selectively permitting the growth of transformants. See, when we are integrating something into the plasmid DNA, it should have the ability to recognize the transformants and non-transformants. Okay, so selectable markers are the markers which identify the transformants and the non-transformant clearly, and it helps to selectively permit or allows the growth of the transformants. Okay. Then, transformation is a procedure through which a piece of DNA is introduced into host bacterium. Okay, generally the genes encoding resistance to the antibiotics such as ampicillin, chloramphenicol, tetracycline or kanamycin are considered to be useful selectable markers of E. coli. Okay, all these resisting genes. Okay, see in the case of plasmid, I told you that is an extra chromosomal circular DNA. In that circular DNA, what is present? Genes are present. In that genes, what is the speciality of the gene? The genes are able to resistant all those particular kind of things, that is antibiotic. It can be resistant to ampicillin. It can be resistant to chloramphenicol. It can be resistant to tetracycline. It can be resistant to kanamycin. Okay, all these resistant gene can be taken. Okay, it can be taken into consideration if those genes have been taken, and at that particular site if something is introduced, then that resistance gene have been lost and the bacteria will be growing. Okay, got it? Understand, it is also very important. The second thing is selectable markers. Okay, now let's see what is next. The cloning site, where should be integrated? Okay, that is another important point. Cloning site. Okay, cloning site. Okay, so what is cloning site? To link the foreign DNA, the vector need to have a single recognition site. What it should have? It should have a single recognition site. Okay, single recognition site for the commonly used restriction enzyme, as the presence of more than one recognition sites within the vector will generate several fragments. Okay, that means one restriction site can give, or what restriction site can give many different fragments which will complicate the gene cloning. If more uh, number of fragments are formed, then it can complicate the gene cloning. If one is present, then it can easily regulate the formation of the different fragments. Okay, so next, the ligation of foreign DNA is carried out by the restriction site present in one or two antibiotic resistant genes. We have already mentioned the antibiotic resistant gene previously. What are the two or three antibiotic resistant genes present? Ampicillin gene is present, ampicillin resistant gene is present, tetracycline resistant gene is present, then chloramphenicol resistant gene is present, kanamycin. Okay, all these are the genes which is present in the bacterial body. That also you have to keep in mind. Okay, and we can say all these can be ligation. Okay, those DNA, foreign DNA have to be ligated in the cloning site, right? That cloning ligation can be done with a restriction site which is present in between the two antibiotic resistant genes. Okay, so that's all about the cloning site. Then vectors for cloning genes in plants and animals, Agrobacterium tumefaciens, okay, is able to deliver a piece of DNA known as TDN. This is in the case of plants. Okay, in the case of plants, you know about BT cotton, right? BT cotton. And also BT brinjal, biotechnologically modified brinjal, biotechnologically modified what brinjal cotton, etc. can be formed. Okay, in that case, the Agrobacterium tumefaciens, that is another case of a dicot plant where that is a pathogen. Agrobacterium tumefaciens is a pathogen, and this pathogen is able to deliver a piece of DNA, and that DNA is known as tDNA to transform normal plant into a tumor plant. Okay, if Agrobacterium tumefaciens is present inside that plant, what happens? A normal cell will be converted into a tumor cells. Okay, and direct these tumor cells to produce the chemical required by the pathogen. If pathogen is introduced into that cell, what happens? Not pathogen, the DNA is introduced into that cells. What happens? The normal cells will be converted into a cancerous cells, and this cancerous cells are able to produce the chemical which is required by the pathogens. Okay, retrovirus in animals have the ability to transform normal cells to cancerous cells. This is also used. Retroviruses are also used in biotechnology. Okay, that's all about the vectors. Then let's see about the structure of a vector. Okay, this is the structure for a vector. What is in the structure? See, you can find a different restriction endonucleases site, right? Ori region you can find here. Okay, what is the function of Ori region? As I have mentioned, what is the function of Ori region? Ori region is a site where replication starts. Okay, then next one, it is important. ROP gene is also important. Okay, next you can find different restriction enzymes. PVU2 is a restriction enzyme, SAL1 is a restriction enzyme, BAM-H1 is a restriction enzyme, HIND3 is a restriction enzyme, CLAV1 is a restriction enzyme, EcoR1 is a restriction enzyme, PVU1 is a restriction enzyme, and PST1 is a restriction enzyme. See, if EcoR1 is given to this, okay, if EcoR1 is given to this plasmid, this EcoR1, it will be cleaved at this particular site. It is, since it is an endonuclease, we can say it will give a sticky end. Okay, so next one, HIND1 and HIND3 and CLAV1. If it is given to this site, here what happens? This site will be created. Okay, what site is created? Sticky ends will be produced. Okay, then this one is BAM-H1. If BAM-H1 is given to this side, what happens? It is cleaved in this side. Okay, so a restriction endonuclease acts upon, and it is given to which, it is present at which region? Tetracycline resistant genes. Or tetracycline resistant genes means if this plasmid which carries the bacteria, plasmid is in the bacteria, right? So if the bacteria is given the medium as tetracycline, if see, if this is a Petri plate, okay, Petri plate, we have inoculated this with the agar-agar, and we have given ampicillin as the medium, ampicillin in the medium. Okay, ampicillin, sorry, tetracycline in the medium. What we have given? We have given tetracycline in the medium. T, I will represent like this. Okay, tetracycline in the medium. What happens if tetracycline, if BAM-H1 comes here and cleaves this one? Okay, tetracycline resistant genes won't be resistant genes won't be acting upon. Okay, that gene will be inactivated. Okay, since that gene is inactivated, the bacterial cells cannot be able to grow. If then this case ampicillin resistant genes, that resistant, if ampicillin is given to the medium, what happens? Ampicillin resistance gene is absent, right? So we can say ampicillin resistance gene is present, so we can say that bacteria will not be able to grow in that medium. So that conditions, dear students, that you have to study. There are chances to ask the question from this portion like that. Okay, with the medium given, and how is this organism can be grown, what organisms can be grown, which kind of organisms can be grown, ampicillin resistant or the tetracycline resistant genes are being grown or not will be the question. Okay, so is it clear about this? Is all about vector and what are the characteristic features for vector? Or I region should be there, then cloning site should be there, then about next we have to study, we have learned about the cloning sites. Okay, cloning site, that are the important points that we have to keep in remember in this case. Okay, so that's all about the vector. Then next we have to study about competent host. Okay, towards which host organism and how the organisms can be go into the host organism. The host organisms, see, the cell is, if you are considering bacteria, bacteria cells DNA, or the DNA which is present in the host is actually lipophilic in its nature. Okay, not lipophilic, hydrophilic in its nature. That means what is hydrophilic molecule? DNA is hydrophilic in nature. Okay, DNA is hydrophilic in nature. What is hydrophilic in nature? Hydrophilic in nature means they are water loving substances. Okay, so if so what happens? the DNA cannot be passed through membranes. Membrane is lipophilic in nature. So since it is lipophilic, hydrophilic molecules cannot be passed through the membrane surface. See, I told we have already learned the structure of membranes, right? They are have bilipid layer. Okay, which is made up of phospholipids, then integral proteins are present, then again the membrane structure. So these they are lipophilic in nature. Okay, hydrophobic in nature. So DNA is hydrophilic in nature. Since DNA is hydrophilic in nature, it is not able to move or pass through the different membranes. Okay, so in order to force bacteria to take up the plasmid, we have to insert the plasmid, right? So what happens? The bacterial cell must be first be made competent to take up the DNA. So the membrane have to be much enough to enough to take the DNA, take up the DNA. How it is done? It is done by treating them with a specific concentration of divalent cation. Okay, so it is these bacterial cells will be treated with a divalent cation. Then what are the cations used? Calcium is used. Okay, which increases the efficiency which DNA enters into the bacterium through pores in its cell wall. Okay, when this see this is cell membrane, right? So it is treated with calciums. Divalent cation, calcium. Calcium is a divalent, Ca2 plus, right? So that is a divalent cation. When it is treated with this divalent cation, what happens? It increases the pore surface. Okay, the pore surface, the pores will be made. What is made? The pores will be made. When pores are made, if DNA is present here, that is DNA. This DNA can be moved into, or the plasmid can be moved inside the cell. Okay, the plasmid can be moved inside the cell. I think it is clear for you. Okay, when recombinant DNA. Okay, so when recombinant DNA can be forced into such cells by incubating the cells with recombinant DNA on ice. Okay, we have a recombinant DNA there. So recombinant DNA can be, see, consider this as recombinant DNA and this as membrane. So this DNA can be integrated into this membrane by placing them briefly for 20 degrees Celsius. Okay, we can give a heat shock. Okay, and also then putting them back into the ice. Okay, we can put them back into the ice. Okay, clear. So this enables the bacteria to take up the recombinant DNA. When it is given heat shock and putting them back into the ice, the bacteria are able to take up the recombinant DNA into their body. Okay, clear, fine. So the next one, it can be done by the two different processes known as micro injection and biologistic or gene gun. This is very important. This is very important. The next part, that every time at least we'll get one more question from this. Okay, either we'll get what a multiple choice question, or from the 2 marks you can expect this one. Okay, so what is micro injection? The recombinant DNA, it is directly injected into the nucleus of animal cell. See, if you are considering the animal cell, it has a plasma membrane. Inside the plasma membrane, it has a cytoplasm. Cytoplasm inside the correct central region, what it has? It possesses a nucleus. Okay, and all the cell organelle will be present here in the cytoplasm. Okay, all the cell organelle will be present in the cytoplasm. So what is micro injection? Micro injection, we have a recombinant DNA. Okay, we have a recombinant DNA present here. We have a recombinant DNA produced. Okay, we have a recombinant DNA produced, and this recombinant DNA produced is directly injected into the nucleus. If it is directly injected into the nucleus, then we will call it as micro injection. Okay, so next one, it is biologistic or gene gun. What is biologistic or gene gun? It is suitable for plants. Condition is plants. Micro injection, that's also one question. Okay, micro injection is given to animal cells, then biologistic or gene gun is given to the plant cell. Okay, so we can say in the case of plant cell, we can say there is a cell wall is present. Okay, inside the cell wall, what is present? The cell membrane is present. Inside the cell membrane, towards the peripheral region, the nucleus is present. Okay, so what is the case of gene gun? It is bombarded with high velocity microparticles of gold or tungsten. Dear students, it is asked in many questions. Okay, what microparticles is added? It is coated with, it is bombarded with micro particles of gold or tungsten. Tungsten. Okay, gold or tungsten. Gold or tungsten. Gold or tungsten. It is bombarded with gold or tungsten. Tungsten with high velocity and coated gold or tungsten coated with DNA. Okay, it is coated with DNA. So if you are considering this is micro DNA. Okay, so not micro DNA, it is the recombinant DNA, and this recombinant DNA is coated with gold or tungsten. This gold or tungsten, gold or tungsten. So this gold or tungsten coated DNA is bombarded to plant cell. Okay, it is bombarded to the plant cell, and this method is known as gene gun method. Okay, first one it was micro injection where the recombinant DNA is directly injected into the nucleus of the animal cell. Okay, whereas in the case of gene, second one it is gene gun or biologic. So what happens there? It is taking place inside the plant cell, then it is directly bombarded to the plant cell with a tungsten or gold plated DNA. Okay, clear? So that is the method how this recombinant DNA is directly injected into both plants and animal cells. Okay, so that's all about today's session. In today's session, we have covered about what is biotechnology, how biotechnology can be defined. Okay, what are the types of biotechnology, and next we have studied about the steps involved in biotechnology process. Okay, recombinant DNA formation, steps involved in recombinant DNA formation. Then we have learned about what are the different tools involved in recombinant DNA technology. Okay, the main thing is restriction enzyme and vector. We have looked into restriction enzyme, types of restriction endonuclease enzymes, that is endonuclease and exonuclease. What is endonuclease and what is exonuclease? It is very important, dear students. Please keep and remember that you have to study that portion, particular portion, and you can leave the home to write the examination. Okay, next you have to study about the next part is um, next part is about last one. We have discussed about vector and what are the characteristic features of vector to be remind. Okay, then later on we have studied what are these two techniques. Okay, micro injection and gene gun technique. Okay, so keep tuned, stay tuned with us for next video. I'll be back in coming days, so you have to be stay tuned. Thank you. Have a nice day.